to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 77th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I've been in the LBA and gene therapy space for over 29, almost 30 years now. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for over 29 years.
John and I are members of our growing scientific advisory team at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio services. Today, we're here with some special guests, Dr. Habibi Goudarzi and Rochelle Burke, both from our Discovery LCMS team. We welcome them, and so we are thrilled to have you listening to the 77th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we may have had from our audience. We're constantly looking for topics, and we'd be happy to discuss something you want us to cover. To kick us off for podcast number 77, John is going to go over today's podcast topic a bit before we jump into the news and resources section. John?
Good morning, Dominic. Good to catch up again. As usual, we'll go through some news items, and then our main topic today is progression of bioanalytical needs for non-GLP studies from early PK to readiness for GLP Tox Studies, with special guests Habibi Goudarzi and Rochelle Burke.
Okay, John, why don't we just jump right in with the news and notes?
Sure. So first topic is actually a nice addition or nice follow-on from what we talked about last week with the GLP-1 analogues. And it's the two big sellers at the moment, Wegovy and Mounjaro, are both administered via one-time injections, and these are plastic-containing vessels, and as a result, there's a lot of concern about the amount of waste generated because they are one-time use only. So actually, the drug companies involved, the actual headlines are how drug makers are handling new pressure to recycle plastic injection pens as millions of new patients begin using GLP-1s. And I think Novo Nordisk, being based in Denmark, are at the forefront of this. And actually, the article talks about how furniture in the Novo Nordisk facility is actually made from recycled drug pen injectors. And so they have a program called, if I could just find it.
Why are you looking that up? That's crazy, right, John? So that literally at Novo Nordisk, their cafeteria chairs and furniture are made from recycled diabetic pens. That's awesome.
I think it's fantastic. Really cool. I mean, I'm big into recycling. I love that kind of stuff. Always looking to recycle whatever I can.
Yeah. And
so, I mean, and I think that a lot of that does come from Europe. We were in a conversation recently with a customer where and this has been a topic within the European Bioanalysis Forum, but trying to make bioanalytical reducing consumption within bioanalytical, and the one conversation was really around the use of monkey matrix and trying to minimize that. But you can see there's this initiative in Europe, and some of this is patient driven. But yeah, I still haven't found the name of it.
You can go ahead and look. I was just
I've got it. The program's called Remed, and so they're relying on people giving their pens back. So at the moment, it's largely patient driven, and they have something like a 50% uptake, and they're aiming to get to 85% return rate by the end of 2024. And as they talk about it, so they have a facility in Europe for doing all the processing. Right now, they're good for Europe because they can transport via trucks, and it means that the footprint of those pens isn't increased by trucking it. But because these pens are used worldwide, so they can't justify shipping plastic, via air back to Denmark to recycle. But it's not just Novo Nordisk. I think, like I say, that's the first one, and that's the one that's furthest advanced. But Lilly and Sanofi are also looking at this kind of thing. Lilly and Sanofi are set up a collaboration in France, and Lilly have also done their own their standalone thing in Germany. So there's nothing in the US yet, but there is obviously. There'll be increased focus on this, and I think this is a great way to go on this.
Yeah, no, it's the recycle pen is the one in France, but it's fascinating stuff, John, to think about how creative they got with using these. And you make a good point about what's the point of collecting them and shipping them all over the world? That's not kind of making it mood. I was just thinking about you're right. There's always it seems like the U.S. is always a little behind Europe and some of this stuff. And I don't know. As a sidebar, the previous weekend, a neighbor and I were talking about how we're trying to reduce our carbon footprint a little bit. Not that I'm, a huge person into that, but I'm thinking about doing more composting, as well as just the sheer number of material you throw away in your garbage bin versus your recycling bin, John. So that was a conversation we had, and he's got a family of 5, and he said he's down to 2 kind of kitchen bags per week on average. And I thought, wow, we're only 3 as a family, and we're probably 1 more than that. So it just got me thinking about it. It's a random sidebar, but I think it's great. I think
with me and my wife, it's just we're good in reducing garbage, but the biggest consumption for us is bags for cat litter. Anyway, to go back to this,
and he had someone, John, he has someone come and pick his compost up. So I mean, he's really it just inspired me a little bit. I thought, man, I definitely want to do more. But back to this, you're right. The GLP world is booming, isn't it? It's just booming.
So basically, this pilot began in 5 Danish cities in 2020 during COVID-19, but it's now a national program in Denmark. It's collected 2.4 million of Novo's pens in Denmark to date, currently with a 50% recycling rate. And like I said, this is aiming for the 85% by the end of the year. Merck have also joined this program. So actually, Eli Lilly, Sanofi, and Merck have joined Novo Nordisk, but they also have their own separate initiatives. And the effort has expanded into UK, Brazil, France, and Japan. So it's gaining momentum. I love this kind of thing. I love it's. I'm fascinated by the whole development process, but then everything you do always has an outcome. And so when you have something like this, where the outcome is a lot of plastic waste, that people have put their minds together and say, how can we address this plastic waste that we're not going to not generate the waste? It's absolutely necessary to treat this patient population, so we have to do something different. I think this is really. I'm really impressed with this.
Yeah, no, it's great engineering. It makes me think, well, why not implants? That's probably where it'll move to eventually. But until that, there are implantable devices that can just shoot you a shot on a they do it for diabetes. I think type 1 diabetics have stuff like that. But it's and then you just have to load the cartridge up, right?
Sure.
So you're already cutting down the footprint there. But John, it's a great way to start off our podcast. It's a wonderful story. Leads it nice to our GLP-1 theme, which, as we know, John, that's not going away anytime soon. And so it's great.
Yeah, I think
that back end, right?
We talked about the elephant in the room last week, and it's going to keep stampeding around every podcast from now onwards. It would be like the new COVID main topic,
Yeah. We'll say news and notes first. We'll start with GLP-1 news, and then the rest of the news, right? Is that what you mean? Yeah.
Yes.
Yeah. No. Well, all right. That's a great one, John. Good way to kick us off.
So this one's really nice and brief, but again, I love the simplicity of it and way people have thought how to address a problem. This is quite quick, but basically, Neurocrine wins approval of Ingrezza sprinkle formulation for patients with trouble swallowing. So they basically Neurocrine Biosciences won approval for a new formulation of Ingrezza to really help patients. It's really addressing a patient need. So the drug is approved for tardive dyskinesia and Huntington's chorea. It was initially approved in 2017 for tardive dyskinesia and Huntington's chorea, but both of those conditions may actually impact a patient's ability to swallow. So it's interesting. We talk about GLP-1s and how there's this move to go to an oral formulation to minimize waste and make it easier for patients, but it doesn't actually always necessarily make it easier for patients. So for Ingrezza, it's actually a capsule formulation, is the way it's, and you basically obviously take capsule, take it with a glass of water, and down it goes. But for this subpopulation who can't swallow properly, that's a challenge. And so this formulation is basically, it's oral granules that are designed to be opened and sprinkled over soft foods, as opposed to the normal capsule. And so, sprinkle it into your food. I don't know if you store it in or what, but obviously because you're limited to soft foods because you're swallowing issue, having the drug in there, then you can get it into your system, and you can have treatment proceed. So it's, like I say, very simple. It's a really nice. We talk about drug delivery quite a lot, but this is a very very sort of what sounds basic, but it addresses a genuine need. I thought this was very is great.
I agree. It's fascinating to think about how Neurocrine is listening to their patients first and foremost, right? That's not always something that it
Sure.
feels like you're, are you being heard? And then what great engineering, really smart people to be able to take an oral pill and say, hey, why not just, I imagine they pop the capsule open, sprinkle it over whatever food as you kind of mentioned, it's not quite clear just how much what they can actually eat, but obviously sprinkling it over whatever it is that you're consuming, even if it's a smoothie or whatnot, you can drop it into there. Fascinating, right, John? What a great thing to help out these patients that are already suffering, and to make their their compliance to their medicines, or even the medicines, more tolerable, is always just a great advancement. And hats off to, I think of scientists, but it's engineering to me, but hats off to them for doing that. And really great stuff, and pretty cool. As basic and as simple as it sounds, right, John? But
yeah, absolutely.
Years of research in there to get it approved, and kind of how to make it into a format that is easily to sprinkle over,
they swallow, and then has the right properties in the body once it's swallowed, and all that kind of stuff. Yeah,
that's the other piece. Yeah, the efficacy of the drug, right? That's the hard part. It's easy to make it into a different dosage or whatever. It's easy to make a different format for it. Is it still effective? And then how do we have to dose it, right? That becomes the kind of I don't know about the challenge, but that's the piece that needs to be researched. So great stuff, John. Really cool. And hats off to the Neurocrine Biosciences people.
So next one up is another follow-up to a discussion we had way back when. UPenn and Stanford analysis finds no secondary cancer cases directly due to CAR-T. And this was something I think popped up a few months ago, where the FDA was talking about adding a warning to CAR-T, CAR-T medications, because there'd been a low incidence of secondary cancers. And so obviously this is basically Carl June at UPenn basically said that they've gone they and the team in Stanford as well have gone through the data and shown that although there has been a small percentage of secondary cancers, none can be actually directly attributed to the CAR-T therapy. So I thought that is a good one to follow up with, and I also thought I'd throw this out for you to
it's great,
CAR-T stuff.
Yeah, it's of course one of my idols, Carl June, right? I still hope to meet him someday. He's an incredible scientist, and kind of the godfather of autologous T cells, or even certainly CAR T cells, right? And John, you're right. There's this concept of like not biodistribution, but oncodistribution is like this new word that's being coined to create a certain degree of what I would call now paranoia within the autologous T cell and CAR T cell world, because there was, as you mentioned, they wanted to halt these life-changing, life-altering, life-saving drugs. And look, we take safety very seriously, so I'm not kind of belittling the industry in any way, shape, or form. It's just this news, right? A 1,500 patient study, some of the really early innovators, they got 20 years of data. What do they show? Like a 2.3% or something? So,
yes, it is.
So patients with cancer are susceptible for other cancers, right? Independent of the treatment. So it's really a great, I did not see this come across my desk over the last couple of weeks. So, when I was preparing for the podcast, reading up on this, it made me feel warm and fuzzy, and really happy to hear that, because it felt like the CAR T cell world, or even the autologous T cell world, or the cell-based gene therapy world, was kind of kind of took a blow from that, right, John?
Absolutely, yeah.
Really, this kind of says, hey, not so fast. And the stats really back up that. Look, with any therapy, there may be some side effects, but they're minimal here, and the gains far outweigh any of the side effects here. So great story, John. Really good that this is coming up.
So basically what happened in January, the companies there's 6 approved CAR Ts, and they were all told to add box warnings at the risk of T-cell cancers to all 6 commercial CAR T-cell therapies. They said, the FDA said in January that they thought 3 cases were related to CAR T, or most likely involved. So it was obviously not proven, but it was like that was their take on it. But at Penn, they looked at 783 patients and 18 patients with secondary cancers, and that's where 2.3% comes from. But none of the cases was there the insertion of the CAR molecule into secondary malignancy, so nothing where you could directly attribute the CAR to a secondary cancer that occurred in a patient after treatment with gene-modified T cells, June said. At Stanford, they looked at 725, so a slightly smaller population, 25 secondary cancers, 11 were solid tumor, 13 were acute myeloid leukemia, and 1 was a T-cell lymphoma, but there was no CAR transgene insertion in that one. So that's probably, obviously, of all the ones, they said that's the biggest concern, where there might be that likely link. But they certainly, based on what you, Penn, and Stanford are saying, there isn't a link. Now, a cynical person might say, well, they might be a little bit biased in proving that. Well, I think this is a discussion that will go on, but I think it's still useful that they've done this initial assessment. And then, obviously, there'll be continued conversation between them and the FDA. And obviously, it's something that people will be aware of and checking for.
Yeah, I mean, I've talked about how the immune system's got this dual-edged sword to it, right? These CAR T cells are incredibly powerful at honing in on tumors, solid tumors, right? Or even blood tumor, any sort of leukemia, lymphoma type treatments. Incredible advancements. John, if those that listen, my first job in 1994, I was a cell processing engineer doing autologous T cells. So this is near and dear to my heart, right? But these numbers are pretty. There's over 1,500 patients between the 2 universities.
Yeah, I agree.
Real low.
It was always acknowledged it was a small subset, but yeah, I think it's an interesting, next step on this.
And it's always something you want to monitor when you put in these power. These are pretty powerful, but like it's unbelievable how tolerant these therapies are, and then they're coming with allogenic, John. So that's going to be, there's some interesting, maybe next podcast. I know there was a couple publications on allogenic T cells or allogenic cell-based gene therapies, which are really going to change. That's like either our kids or our grandchildren will be able to get treated. Hopefully, like, going to the hospital, something's wrong, boom, you got cells in a bottle for you versus this process. It's like a couple, probably takes, I think, anywhere from 4 to 8 weeks to get these CAR T cells engineered for you. So that's, that's a little off topic to this, but it's a great story, John. Love that the innovator, the godfather of CAR T cells, Carl June, kind of published this. But, John, I got nothing else on that one, because the next next story you're going to talk about, I did read about this one, and it's a good one. I like
this one. Is really fascinating. Now, this is, again, we love to sort of say, where are we going in the future? Here's some of it. This is mRNA vaccine for glioblastoma targets tumors in humans and dogs. And this was published early May in Cell, and it's researchers from University of Florida and a spin-out biotech called Ioncology. They've got 7 years worth of experiments culminating in a first of its kind phase 1 trial in 4 patients and 10 pet dogs who developed brain cancer spontaneously. The human patients survived or lived without disease for longer than expected, according to a press release, although it noted that it's too early in the trial to analyse the vaccine's clinical effects. Meanwhile, the dogs lived for a median of 139 days, far longer than the typical 30 to 60 days survival. So this is glioblastoma is a very aggressive and usually fatal. So this was, I don't know if you've seen Eric Tophol's, he's at Scripps and appears on Twitter and various places, really sort of said, after marked success treating 10 dogs, the vaccine induced a striking anti-tumor response. So this is like a completely different approach to doing this. And I'll let you talk, and then we'll
Yeah,
maybe touch on the structure,
sure.
etc.
And I won't steal your thunder because
No, no,
it's
it's fine.
incredibly fascinating how they engineered this, right? But just glioblastoma is, as you mentioned, it's aggressive, and they're over the last about, I think it's been almost 20 years, but at least maybe about 15. There were autologous T cells for treatment of glioblastoma as well, before CAR T.
Hmm.
They were trying to perfuse it into the spinal cord or maybe directly into the brain, right? All sorts of kind of stuff that's that seems evasive to me, invasive, or yeah, I think that's right, to the patient, and just very expensive and kind of hard to do. But these mRNAs, John, just similar to CAR T, but mRNA technologies are, man, they're just exploding. I know we think of it for COVID, but they've been around for a good couple decades prior to the mRNA vaccines for treatment of COVID. So these are not new for treating immuno-oncology and other sorts of rare diseases. But John, this is great stuff, right? Do you want to dive into how
Yeah,
they take advantage of the microenvironment? It is fast. This is
if you don't mind,
I know you tell us about it, but
I'll
got
do
me
something.
excited. Yeah.
Yeah, so basically, obviously, you hear a lot about cancer vaccines and big challenges. How can you stimulate a response? And I think that has been the challenge to date. Typically, with mRNA, they're basically in a lipid nanoparticle that target toll-like receptors on immune cells, and that's really how the mRNA-based COVID-19 vaccines were based, and you'd basically train the immune system to react to a disease, but isn't targeted to a specific patient. And these ones, they structure they use RNA lipid particle aggregates, so they actually pull several lipid nanoparticles together to make the artificial structure look like a virus. So that engages a protein called RIGI in stromal cells, a type of stem cells found in tissues throughout the body, as well as the tumor microenvironment. When injected into the bloodstream, this stimulates the release of a large number of cytokines and chemokines, along with dendritic cells and lymphocytes, training the immune system to go after the patient's cancer. So it's a completely new approach, and but like I said, that this really does elicit a strong immune response. Christina von Rummeling and John Ligon at University of Florida both said in this in a separate article to the one in Cell. So I basically said they inject these systemically rather than directly into skin muscle or tumor itself, and that perpetuates the effect. So I'll pass it back to you, but
Yeah.
it's
It's
great.
amazing. Yeah, it's amazing. So even so much to unpack here, right? So like
Yeah,
the way the LNP is engineered is onion-like. I think that's important to
yeah.
stress, right?
I didn't quite
And then,
understand the onion like.
yeah, it's hard to discern.
I,
Yeah,
know, I get when they're saying onion peeling away the layers, but then it mentioned about the onion bag, and
it,
it's like, yeah, I think we need a better description than that.
it
But
does.
that aside,
Yeah, and John, when normally when you start to form immune complexes, because that's a safety issue, but apparently
sure, yeah,
these
yeah,
don't. So it must be, this article doesn't tell exactly, but it's just what it's really doing is amplifying the message. Really, it's like creating this
sure.
like massive warhead, is how I think of it. But as the other, as you kind of alluded to, the traditional mRNAs and LNPs take advantage of innate immune kind of processes, pattern recognition, all sorts of toll-like receptors, really kind of that innate immunity kind of triggers. Well, this very, very wisely, right? Similar to what we see with certain conjugation technologies that don't become activated until they're in the tumor microenvironment. So these mRNA people probably went, oh, why don't we try something like that? And sure enough, they looked for this protein in the tumor, the stromal cells, right? Is at that RIG1, I guess, right?
Sure. Yeah.
And so what does it do? It must have some sort of way to traffic into the tumor microenvironment, hit that and get activated only when, instead of, because toll-like receptors are ubiquitous, right? So this, and obviously cancers have maybe a higher rate. They try to take advantage of that. But this is a tumor-specific stromal type of protein that it's targeting, and boom, sure enough, this is, and then, as you kind of spoke about the the wave of immunological responses is now like targeted to the tumor, right? Right. All the things that you're looking for. It really, this is fascinating. Hats off to the U of F uh, scientists. I don't know if they're down in Alachea there or Alachea. I don't know. That's down in Florida. I don't know if I'm saying that right, but man, this is great stuff, John. I mean, the advancement of this is incredible. I know it's only in dogs, but they talk about just the concept of a cold tumor that's impenetrable within 48 hours becomes hot.
Yes,
That,
exactly.
I've been trying to, John, that I've studied extensively. Even so, as I alluded to, we knew we were onto something with autologous T cells in the 90s, but we couldn't overcome that tumor microenvironment. We
Sure.
talked about
Yep.
it. It was all about trying to get dendritic cells. And remember, dendritic cells weren't well studied. They're only like 25 years in the making, right? I mean, they've been in our body forever, but we didn't have tools to really analyze them until about 25 years ago, right? Flow cytometric things. And maybe you could go back a little further, but I think you know what I'm saying. And now here we are today with like the ability to turn a cold tumor, because that's still very hard to do, meaning you can give an animal a tumor and then some sort of chemical cell line it and cure it with a number of different therapies. But if there's an animal that naturally has the tumor, a cold
Sure.
tumor,
Yeah.
that is still very hard to cure. This
Yeah.
seems to have been able to overcome it, and that's why the excitement's there. So great stuff, John. Just really hats off to UF for this. It's fascinating.
Yeah, and yeah, absolutely. I echo every word you said. The next state, obviously, they see the potential and they want to go with it. So they're now at the stage of establishing dosing, and then they're looking to enroll 25 pediatric patients for phase 2. So they also hope to test the vaccine alongside other immunotherapies, as early evidence from their experiments suggests a combination approach might be worth pursuing. So again, it's like with so many of these things, we'll keep watching for
Yeah.
this, because this is like the first we heard of it, and
Yeah.
it's amazing. So yeah, it'd be
Think
really
about this, right? We can move on to our guests. I'm sure they're waiting with bated breath to start our main portion here. But what about the cost of this relative to a CAR T or
Mhm.
other therapeutics? These are
Sure.
very affordable, and that's the excitement, I think.
Oh, sure, yeah.
As indicated by billions of vaccines that we produced, this could be a way to treat every cancer patient worldwide. And of course, we love that, don't we, John? That is a
Sure.
goal. So
Absolutely,
great
yeah. I'm all in favor.
yeah. John, yeah, no, great news and resources section. I don't know if you want to introduce our guests, but why don't you go ahead and tell us what we're going to talk about, and we'll get.
So yeah, so we like to try and involve as many other people from the company as possible, and today it's the turn of our non-GLP LCMS group, and we actually thought as a topic we'd say progression of bioanalytical needs for non-GLP studies from early PK to readiness for GLP Tox Studies. And we've got Habibi Goudarzi and Rochelle Burke with us. We both lead the team. We should have had Cassie Manning as well, so we're going to have 3 people. But Cassie, congratulations to her, is actually going into hospital to have her second child. So it's only 2 guests rather than the 3 we were expecting. But I think let's just start with an introduction to the team. And Habibi, do you want to go first? Talk about yourself, give a bit of background, and then we'll move to Rochelle.
Yes, I can. Thank you. I've been with KCAS for 6 years now. This is my year 7, but about 20, 30 years before coming here, I've been involved in all types of assays projects in life sciences area, focusing a lot on mass spectrometry. I've been doing mass spectrometry since 1986, so it's been a lot of years and all sorts of different type of instrument types, type of experiments, etc. So I think it was a good training grounds to get me ready for coming here and starting a new group and watching it grow.
So, what was the first mass spectrometry you worked on?
It was a custom made, which was a sector hybrid instrument. It was a permanent magnet and a quadrupole.
Okay.
It was used for surface dissociation. That's when you select the ion, smash it into a stainless steel small surface, and then try to extract the fragments off of it. So There's a whole bunch of different ways of putting energy in a molecule, make it fragment. So that was it. And then I modify it into add another quadrupole and did charge exchange type of um, work.
Okay. Yeah, yeah, because that's similar sort of time as when I started, and so
Yeah.
we had a Finnegan 4500, I think it was.
Yep.
That was a single quadrupole, and obviously Finnegan eventually became part of Thermo, and then in another room we had a VG 7070.
Yep.
And that was the typical magnetic sector instrument, and that VG then eventually became Waters, so both
Yep.
companies still in existence. Anyway, let's move on.
Yeah, why don't we let Rachel introduce herself?
Enough of the nostalgia
You
piece.
are as old as dirt with these instruments you're talking about. No, I'm just kidding. Rochelle, well, why don't you say hello?
Yes, here I am. I'm Rochelle, and I have been here at KCAS Bio for over 16 years now. And actually, my career started before I got here at KCAS. I started in the environmental industry and then moved into more like drug screening. And actually, both of those jobs I had were GCMS focused, and it wasn't until I got here at KCAS that I was introduced to LCMS. Majority of the time that I've been here, I've done pharma work and then I did some AH work. So my knowledge and expertise is mostly in the pharma area. And I've learned a lot since I've been here. I've learned how to do the GLP regulated side. And then the last 6 years, I've learned how to do method development under Habibi. And so my knowledge has advanced significantly since I've been at KCAS.
Yeah, thanks, Rochelle. That's great. So you've made that journey from GC mass spec into the regulated mass spec world, which we do a lot of here, and then you've now in the last 6 years, if I understood you, you've kind of fallen under the mentorship of Habibi here, and you guys do all things non-GLP, and boy, do you do a lot in that department. So, John, I kind of just jumped in there, but I know you're
Sure, go ahead. No, let's
on the agenda. And
conversation.
so, yeah, I don't, and when I say a lot, it is that your team. I often refer to you guys as the Marines. I know Habibi has heard me call you all that, meaning we talk about like what is non-GLP, but to me, I describe it as intact small molecules, oligos, peptides. You guys can even do some degree of, some of the larger stuff, although I think we have another team that you can kind of cross over with. But typically anything that requires an affinity capture, you might stay away from, although I'm sure you're experts at it. But why don't we just go into that? Why don't you tell us a little bit? Go ahead, John. You jump in.
I was going to say, what do you consider to be your team's brief? Let's start there, and then we can go into more specifics.
Yeah. So one of the actually what's interesting is that the two superstars in that team, Rochelle and Cassie, both have had GLP experience for many years. So even though it is a non-GLP team, they try to follow GLP processes as much as possible without.
Yeah, that's really important, isn't it, Habibi, right? That's important to emphasize because that's the
Right.
whole process of what you all are doing. But I didn't mean to interrupt you too much there, but that background they have, as well as yourself, is critical to the success of our non-GLP team.
Yes. So it's the big. I mean, the reality is that a lot of the methods, assays that the team works on finds its way to regulated work. So we don't want to have we want to have as smooth as possible process that information can flow from one team to the other and the work can continue. So that's one of our advantage. So the one of the standout feature of the team is Rochelle has done her best to put together a team that doesn't have a bottleneck, meaning that everybody learns everything. You guys have heard my term or phrase about powder to report. So it's not segmented. What some people just do sample prep, and some people just do analysis. So everybody learns from start to finish. So there's no. So if you're ready to do method development, you're not waiting on somebody else. You can do it yourself. So that has worked in terms of everybody learning everything and also working in real time, meaning that as soon as data is available, whoever is leading the assay will write the report, and they can also interface with the clients and present their own work. So that has really worked, and it's a standout for the group that we're able to do rapid turnaround. Uh, have worked with some very difficult um, um, molecules or assays, and the only thing that the team does not do is bottom-up proteomic, and that's by design, as you mentioned, Dominic, that we have another team that does that.
Yes, so I think it's important to kind of say, hey, it's some of this is discovery PK work, right, Habibi and Rochelle, and then some of it's like pharmacodynamic and biomarker work too. I don't know if you want to touch on that a little bit, but like you guys do a lot of uh, work on that mass spec and solve a lot of problems for clients. As a ligand binding assay guy, this mass spec stuff is still, I don't want to call it new, but it's a wide open world for a whole array of things that we don't want to touch by LBA, certain cutoff weights and things like that. Yeah, go ahead.
It's the truth machine, Dominic.
Is that it? I still don't understand why you want to beat the heck out of stuff, light it on fire, put electricity through it, and do all these mean things to it. But no, I do think, I kind of, all kidding aside, maybe we'll continue with our agenda, John, and
Sure.
Habibi and Rochelle. Maybe tell us a little bit about
I think it's like you touched on it, that discovery PK, and this is where we're talking the very early. So I mean, actually, I suppose, Habib, you could talk about the range where you start from very early discovery all the way through to pre GLP, and the different needs for that. I view the early PK as speed is almost the most important thing there. Obviously, you want to have a quality assay, but you need to get data. So how do you gear your processes to switch between those need for speed assays and then the others that need a bit more background, method development, etc. And
it's a really collaboration between us and the client. So they may want to do a screening assay and relax the acceptance criteria. We could talk with them, and if we think that maybe a more say qualified assay will be better, we listen to them what decisions they want to make off of the data, and that's the important part. That depends on what you want to do with it. You need to make a right decision in terms of which way to go. So it all comes down to what change in the data will be significant. So if you're looking for 2-folds, 10-folds, 20-folds difference with whatever experiments you're designing, then a screen assay may be just fine. But if 2% difference makes a huge difference in your conclusions, then you want to go with a tighter acceptance criteria and a qualified. And the qualified assays in our world could be as little as 4 different basic experiments to the full suite of what you would do under GLP. It's just a matter of how you do the design the qualification. So again, that's also part of discussing with the client and then understanding what really would be best for them. And we advise, and they effectively have the final say, and they can select what they want. And our role is to advise that. So going back to the type of molecules, we did a lot of assays with the NAD metabolome, with ATP. They were really tough assays, and the team also does. You mentioned PK, but PK and TK parameter modeling. I know the bioanalytical part of it is often referred to as PK, but the PK parameter modeling is also the team does that. So you can go from start to finish with the team, with as many as different things that loaded in the whole process.
Yeah, it's a great discussion. I'm going to turn my question to Rochelle. When we talk about speed and turnaround, and I don't like quick and dirty. I like rough and ready. But Rochelle, Habibi kind of talked about it, or the way the team's set up. Could you share with us a little bit about what are some of the when it works? What's the time? What's sort of the timing that you can get from powder to report? Maybe talk about that, and then what are some of your maybe challenges? But just walk us through a little bit about how you get the need for speed and how you achieve that, Rochelle.
Well, one of the ways that we achieve that is like what Habibi was saying, where everybody knows how to do everything. So we don't just have like one team that does method development and one team that runs samples. Everybody learns how to do everything. So that allows me to give projects to each person. So we can have maybe like six, seven, eight projects going on at the exact same time because each person has maybe more than one project. So that's one way that we can kind of like keep up with the speed and the demand. Most of the time, I try and make it to where like what I call like a rolling backlog where I'm always doing everything in my queue and getting it off my plate like every month, and then I hope to have more projects come on my plate for the following month because most of our clients, as Habibi said, they want stuff done right away. So we have to work with them to meet their timelines. And in this, area of science, they want it really fast because they're trying to make decisions for their next drug or, their next formulation. So we have to try and keep up with that. But also at the same time, I've set up certain processes in my team to try and keep a certain amount of quality. We just have a little bit more flexibility than the
Mm.
GLP teams have, but we still have to like try and do quality and speed at the same time, and it's kind of like a fine line. So we all just work together to try and like make that happen and meet all of our client timelines.
Yeah, no. So you're like interchangeable parts, right? Next person up, but everybody, you kind of, I don't know how much you know about Southwest, but all their planes are identical, or at least they were, except for the number of rows, right? Some planes might have had 100, some had 200, and this allowed for crews and pilots to seamlessly make their way. So if a pilot didn't show up, any pilot could fly that plane. And so that's kind of like your mantra, right? Like you guys have like a templated process. Is that accurate? So that everybody can jump into a project? Is that kind of how you manage some of that?
Absolutely. Yeah. So if somebody is like working on a project, like it was kind of their project, and say they have to take off or something happens,
Mm.
I can easily have somebody come in and take over. And we do that all the time. Like we have people fill in and help out where the need is at in the
Yeah,
group.
and sometimes a day makes a difference, right? You're literally
Yeah.
people are waiting to dose people, and so I know the flexibility and nimbleness of your team is off the charts, and that shows through. Our clients really appreciate that. Um, you touched on something else. You mentioned that quality piece. So that one, right? There's an expectation, right? As John and I are on the commercial side, oftentimes people will be like, "Yeah, we just need it rough and ready, whatever." And then they get into, "Well, hey, did you look at this? And did you look at that?"
Mhm.
I don't know if Habibi or Rochelle, if you want to talk
Yeah.
about some of those slippery slopes, right?
I,
That's probably the biggest challenge for you all, right?
can I start on that, Habibi?
Yeah,
Go for it.
absolutely. Yeah.
Well, I
Go for it.
think that it's really challenging for us because on the GLP side, there's no way to do it except for the GLP way. Like you have to follow certain guidelines. The issue that we have is that we have flexibility, which means that we can change things. And so, like Habibi said, we have to constantly talk to our clients and figure out like what areas we can be flexible on and which ones we can't because we have to guide them because we know a lot of things that they don't know, that it's like if you don't do it a certain way, you are going to sacrifice your quality, which then means your data is going to be sacrificed. So it's a real fine line of trying to figure out how can we get them what they want at the best quality while still maintaining our speed. And it just makes it really challenging because like I said, you don't have to follow the GLP rules, but in some ways you want to.
Yeah. And when
And
you say
no,
that, sorry,
no
go
one
ahead.
wants,
Go
no
ahead.
one really in their heart of hearts wants it rough and ready. They still want a quality product, regardless of the step you're looking at.
Yeah, no, Habibi,
Absolutely.
please jump in and add more, Habibi. Yeah, go ahead.
Yeah, so I want to add something that might not have heard about it, but I think documentation is the key to quality. Even if you make a mistake, if you have things documented properly, you can always go back and look to see, when the data, when it doesn't make sense or is anomalous, then you can go back and, look at that, your documentation. So the type of documentation that the process that Rochelle has for her team, it really speaks volume. When I look at it, is the whole idea of being able to get somebody else to jump in if we need to add resources or cover somebody that cannot be coming to work, and we need to follow the work anyway. if the documentation is not there, the other person would not know what to do, where to start.
Yeah, Habibi, you kind of read my mind a little bit. I think that's why it's important to emphasize that Rochelle and Cassie come from this GLP environment, right?
Right.
And oftentimes it's hard because you only know one way. You're going to document everything, right, Rochelle? You're going to do it as if it's going to go through our GLP, all our instrument. You have no non-GLP instrumentation. The challenge sometimes, though, is a client will say, I don't want to pay for that.
Yeah.
Right? And when I say pay for that, they mean, oh, well, I don't need all that, right? And then what happens? They eventually do want that, though, right? As John kind of alluded to. So great discussion here. Go ahead.
Sorry to jump in.
Yep.
One thing that actually buys us time is we document effectively the same way, but it's all electronic. We don't copy the date, the information, and then have to sign and date the copied paper is one of the expected process steps. Just to be able to quickly save things in an electronic format and continue, that really saves a lot of time as far as documentation goes. So we
You
give
still have peer review QC though, right? You still go through a QC process, right, with all your data. So it's not like the quality's lower.
we call it peer review. So the person who does it reviews first. There's a peer review, and then there's a higher level, call it management or PI review. So we have all that stuff, but we do it internal to the team, so we don't have to wait in queue if you had to send it to a formal QC team.
Yeah, so, and Habibi, that's a whole nother level of scoping, right? If somebody wants a QC review through our QC team, then it's really not discovery work anymore, right?
Yeah.
So that's a different kind of.
Well, QA is where it's really.
Yeah, that's a whole different ball game, right?
Yeah.
We can still do it. It's just what they want. So some clients actually do want a formal review, and we do that. After we get done with ours, we send it through, and
if you have a good BD team, that means you should be getting paid for that. But let's we can talk about that at another time, right? I don't know. So like I want to follow our agenda because I do think the next item, if we had any more about the speed and the type of work, Habibi and Rochelle, you can add a little bit more before we go into our next question to you.
No, we can move on.
Okay, great. According to our agenda, we got challenges with tissue analysis. At KCAS Bio, we got a 45+ year history here, right? You'd be hard pressed to find a tissue type and a species type that we haven't worked with. And there are some challenges here. I think this is one of many things that we solve at KCAS Bio. What helps separate us from our competition is our ability to work in tissues. And it's up to Rochelle or Habibi. You guys can, either one of you can jump in, but tell us about the depth that you have with tissues. And it is a lot of depth.
Rochelle, go ahead.
Sure. Yeah, we work with a lot of tissues and some that people don't even think about off the top of their head. Like we've done like things with eyes, we've done nails, we've done skin. So we're kind of limitless because like you said, we've done it on so many different tissues. Like I'd say over half of our work is tissue work. When we do it in rodents, we do it in cynos. So we kind of know like what kind of tissue, like what size we're going to get. So we know what kind of dilution factor. We know that tissues create challenges. So a lot of times clients will try and say that we should use like a surrogate, like plasma or something. We've done it enough times that we know that that's not the best approach because most of the time you spend more time trying to get them to match versus just using matrix match to begin with. So we already have these experiences and we know brain, for instance, tends to be a little bit more dirty, challenging. So we've been through a lot of that. And like I said, we've done thousands upon thousands of tissues for different clients and different contracts. So we pretty much got it to a well-oiled machine at this point.
Although to go back to what we mentioned earlier, I mean, I agree matrix matching is where you might want to be, but you have to be prepared that, if there's, for example, that initiative from Europe to sort of minimize use of certain species, matrices, etc., that you may have to find what's the best non-matrix match approach that you can take to address that kind of situation. I think that'll become more of a challenge.
Yeah, but still, I think, Rochelle, you said it really nicely about your dilution factor, and that is saving clients time and money. That knowledge, that tribal knowledge, whatever you want to call it, the knowledge we have in the tissue space, and it'll lead into our next item, which is like LNPs, and a lot of it, you're doing a lot of biodistribution, right? That's a lot of what this is in. And so that knowledge around knowing how to like whether it is an artificial matrix, and if we don't have to do that, you are saving time and money significantly by saying, hey, we can anticipate how big the tissue is, and we can anticipate how much we want to dilute it in order to get the sensitivity, the desired sensitivity, right? The desired lower limit of quantitation. So that is the reason why people choose us for these types
Yeah.
of studies. So well done.
And if
Yeah,
I could step in, I mean, I think that experience and that depth of knowledge of working with tissues really adds to the versatility of the group. I mean, I think that is a huge selling point when we're out with talking to customers, and we obviously always talk about all the teams we have, but when you sort of talk about the depth of tissue experience and what we've done, it can make a big difference in terms of winning non-GLP work.
it's the time and somewhat the money, but it's definitely the time saver. And I don't know if we had another Habibi, if you had something to add there, but it was a great discussion about tissues, and I think it's a good segue into really where we're seeing a lot of demand, and that's in the LNP space. Before we get off tissues, please add.
Should we go to larger molecules rather than the LMPs?
Oh, my bad. We'll go on to large molecules first, but
Because that is where the trend is going, and let's go there.
I can start talking about the larger molecules. I saw this trend coming a long time ago, about 15, 20 years ago, that larger molecules by nature, they don't want to stay in a gas phase. So mass spectrometry has to have the molecule in the gas phase before it can be ionized and all that. Well, there's some techniques that you start from solid, but once the electrospray ionization came about, it just opened up the world for the biological significant molecules. So it just as instruments, as mass spectrometers improve in performance and robustness, then it just allows us to be able to just keep pushing the envelope. Right now, I would say to do intact work, the molecular weight's around 10 to maybe even 15,000 Daltons is doable. You just have you're fighting sensitivity, you're fighting the lower limit of quantitation.
Habibi, do you want to say what you mean by intact work? We know what it is, but do you want to just sort of
So if you get a molecule that's 10,000 molecular weight, obviously for a lot of the triple quad systems, that molecule is too large for the mass range. If you can ionize it, get the charge states higher, and you can bring that M over Z or mass to charge ratio to the level where the instrument can see it. So you're not fragmenting, you're not digesting, you're not breaking down the molecule. You're looking at, let's say, a mid-sized peptide, let's say 7,500 molecular weight. You're looking at it intact molecule. You're looking at the molecule in a native form. So you're ionizing the parent compound. Non-intact, where bottom-up proteomics is, takes advantage of that, is where you typically digest with an enzyme to turn it into basic peptides, and then now you have low molecular weight ions that you're looking at. So it's not a big, huge molecule. So some of the intact molecules could even go, let's say, 150 kilodaltons. You can look at it with a time of flight system or other devices, as long as the charge state is high enough. But typically for full quantitation, the sweet spot for what we call low molecular weight, high molecular weight is that 10,000 Dalton mark.
Oh, I'm sorry. I fell asleep. Did you say you use a capture antibody to detect 150? No, I'm just kidding. That was no, great stuff, Habibi. And one of your favorite words that you've coined is a mixed mode macromolecule too, right? Yep. That's kind of in this space. But I am half joking when I talk about a capture antibody to detect, because 150 KD is basically a MAB, right?
Yep.
And so you can definitely, it's an incredible advancement of mass spectrometry or high res mass spec to be able to do those things intact. It's fascinating that you can work around having to get obviously I'm still a little biased, right? Like an anti-id or some sort of like antibody against the monoclonal, or even a capture antibody still has a lot of utility, and they're never going to be. I think the whole field has come to the conclusion it's not like mass spec or affinity capture or whatever it might be is going to replace ligand binding assays or vice versa. It's all about just solving the problem. So whatever makes sense. And that's
Yeah.
our approach at KCAS.
No,
Exactly.
I agree. I've been in analytical chemistry for a long time. So what I've seen is that a lot, essentially, there are many, many different ways of doing things, and they're complementary. So you
Yeah,
get more information from,
exactly. But
yeah,
why reinvent the wheel if I can buy an ELISA kit for something, or there's a capture antibody readily available, and
exactly.
there's plenty of real estate on the compound to get a antibody pair that can be ironed out for
Yeah.
a ligand binding assay. And likewise, it doesn't make sense to have to do any sort of mass spectrometry work unless there is that promiscuity, right? That's the power of the mass spec to me. There's oftentimes a lot of antibodies that just have cross reactivity, or they just don't give you the type of specificity that you really need. And that's when, to me, a mass spec tends to make more sense. Or, there are some generic methods, there are some off the shelf things that a mass spec, if you got the mass spec and you need the method and it's pretty affordable to do and easy to do, then do it, right? Why would I go out and buy a plate reader if I don't need one and I've got a mass spec? So that dictates things quite a bit.
However, go back to the topic, because this is a real topic, and it's worth digging into.
You're killing me, John. I told you I fell asleep when you were talking about that. No, I'm just kidding. Go ahead.
It's yes, obviously the improvement in technology of mass spec has made it possible to work with these molecules up to 10,000 molecular weight intact, but they're more challenging to work with than your typical small molecule. Not saying that small molecules can't be challenging, but they do have their own inherent challenges. Ultimately, the basic flow path of extraction chromatography mass spec hasn't changed. There's been tweaks to the technology we use within that, but we still have a problem. They require expertise to successfully develop assays for larger molecules. So I think that's worth touching on if Habibi wants to, and Rochelle want to talk about what their experience has been in terms of working with these larger entities and the kind of challenges and issues they've had to resolve.
Yeah, I can go first, and then Rochelle can see if she wants to add some more. The Dominic mentioned that the mixed mode macromolecules. So one of the trends is to take a peptide and modify it, and the new characterization or characteristics that the molecule has is different from just being a straight peptide. So one of the basic things, carryover. It's just going to be sticky, we call it, and it requires time to figure out how to lower the lower limit of quantitation and not have carryover that essentially nullifies that value.
So to dig into the carryover piece, sorry to interrupt. Basically, what we do is we develop assays to quantitate molecules.
Right.
If you have carryover, it's basically it's sample, for want of a better word, ghosting from one injection to the next.
Right.
And so it affects your quantitation, particularly for lower concentration samples that can give an artificially high concentration. So then you're giving false numbers, which is really what we want to guard against. I'll punt it back to you.
Correct. So yeah. So and then the other part of the challenge is just the straight math. People that are used to dealing with low molecular weight ions, let's say 500, and then you have a molecule that is 5,000. Well, right there you have 10 times less molecule when you go to the larger compound. So often we don't think about that and don't translate it into, okay, what does this mean? If you because a lot of times we're used to dealing with weights, like nanogram per mil. Well, 1 nanogram per mil for low molecular weight ion is totally different than 1 nanogram per mil for a larger molecule. So that's part of it. Is our challenge is essentially driving the lower limit of quantitation to where the will be meaningful for the experiments that are being planned. And then part of it, as we mentioned just now, that dealing with carryover is a huge deal. We have to take care of it before we can lower that LOQ.
And part of the issue of resolving carryover is where it occurs in the system and how you then address that.
Yep, that's right. So we have experiments that we designed to identify where it's coming from, including if it's coming from the sample path or the column. Like we go through and repeat the LC part without injecting a new sample on column, and we keep collecting data. So if you see anything leach out, then that means it was stuck to either active sites on the sample path or maybe the column itself, and then we design experiments to see how we can wash it out. So that's we've spent a lot of time, so there's a lot of depth in terms of dealing with that problem.
I'm sure there is. It sounds like it could be its own podcast, but we're coming up on top. We got some more time, but we got a couple more topics. I want to turn to Rochelle here. Rochelle, it's your call. You can either add to the large molecule discussion, or we can move on to LNPs. Do you have a preference there, Rochelle?
We can move on. That's fine.
Yeah, I agree. Because you're supposed to do large molecules by ligand binding assay, Rochelle. If you hadn't heard, right?
Oh yeah.
No.
Only if you're a Philistine or you're in the Stone Age.
Oh, is that? I think of myself as more a Greek, kind of like a Spartan. But I got you. No. So Rochelle, right? These LNPs. I imagine you've been working with them forever, right? These aren't new, but boy, I feel like they're really exploding. We heard a great news article about it. You can go anywhere you want here. The floor is yours. You can tell us about your experience with them, thoughts on them, challenges with them, how you can get them rough and ready, how you do biodistribution. The floor is yours, Rochelle.
Well, we worked on a pretty large study with a pretty major client that actually has had some significant news, I guess you could say.
The success, right? Treatment of patients, right? They had a lot of success. Correct. Yeah.
Yes, and so that's exciting that we were part of that, and we did do a lot of biodistribution studies on that. It had very similar challenges to what we were talking about in the large molecules, where it has a lot of like non-specific binding that we had to deal with. So it wasn't exactly easy. And then when you add tissues on top of that, I mean, tissues have their own issues as we talked about earlier. So you're basically compounding your issues, but obviously there's a way, and we figured out a way. And so our method that we had, at least for that client, was very successful. And that's one of the projects that we did thousands of tissues and across like 6 to 8 different projects for them. So we were definitely a part of their success. And so that's something that I'm really proud of. And we had another project that we worked on for another client that has been successful, and hopefully we'll get more work from those people that were in charge of that particular project.
That's great, though, isn't it? It feels good to be able to contribute to something like that. It's a very altruistic feeling, knowing that all the time in the lab. I'm sure there was I know what you're talking about. The whole organization was familiar with this endeavor, or at least, some of us were on just how vast this study was, how big it was, how fast you and the team turned it. And then, fast forward 6-8 months later, boom, you're reading about it and you're like, man, I think I was part of that, right? That's a wonderful feeling, right?
And hopefully you'll get to the stage in a few years' time when it's approved and you know that you were part of that. That's the big one.
When you're watching a commercial or something, you're like, I worked on that. That's the great feeling, right? But no, Rochelle, please, any other thoughts on LMPs? But you're it's not a pressure situation. It was just, I imagine at the time you probably didn't quite realize the vastness of what you were doing, but maybe talk a little bit about some challenges with LNPs, or even like, I know a lot of that was tissue. Just walk us through a little bit around some of the expertise you have.
Well, we had a lot of variances when we were trying to make our calibration standards. And so that was a challenge. We couldn't figure out why, you could put it in. It was almost like there was a cap. So like you couldn't go as high as you need to go. So it was like what's that called? The dynamic range kind of had to be limited because you couldn't go too high, or you started seeing erratic data. And then the same with low. It was like there was a cap there. So we had to be creative and try and figure out like how we could extract it and get things to be, the accuracy to be better, because we were having that challenge in the beginning. And we spent a long, long time on this one particular molecule because of that. Because if you can't get your calibration curve to be within your percent accuracies and to be linear, then you really can't move forward if you can't get past that step.
Yeah,
so
that's dynamic range, right? So this is something we experience a lot, right? Where you got a short dynamic range and then you got to dilute the sample. Did you have to do some of that into the range, or how did that work? Like I understand, like our audience and myself, we understand what you're saying, but that is exactly why people come to us for that type of knowledge, Rochelle.
Yeah, and we actually ended up figuring out too that we used what is it called? I call surfactants Habibi, remind me.
Yeah, the surfactants fine. Yeah, the unfriendly MS molecules.
Basically it's like soap or whatever, but which
Yeah,
is not, yeah, it's not friendly for mass spec. So we actually use that mostly not for like non-specific binding, but we ended up finding that there was like chemistry that was going on that was helping this molecule to basically be stable and limit the erraticness that we were seeing in our data. So that was one of the things that we like stumbled upon when we started doing that, and our method ended up using that, and that was the same method that the GLP team used when they went into their validation. So,
Yeah, so I can add some general comments. Is that when you have a really wide dynamic range, it tests your chemistry, analytical chemistry, and biology knowledge. That understanding how to keep different molecules in solution and be able to have reproducible extraction, good calibration curve, etc. So it was a really strong effort that I'm glad that the client wanted to have a robust assay, and we worked on it with them. And I know it's one of the perception for the team is we do everything fast, but we also do slow stuff too. I mean projects that take more than a week to work on, more than 2 days. So assays could be developed as fast as 2 days, and at the upper end is depending on what we're trying to accomplish with the assay. It could be
Mm.
months, and there are many projects that for good reasons, our clients wanted us to, keep pushing the envelope and get a, better assay for them, and we did. So we don't abandon anything. We just say, here's what we have, and we discuss to see what the next step should be.
Yeah, so maybe I should call you guys Marines and police then, right? No, I'm just kidding. Right? So, because it is, we do think of it as like quick and rapid, but you're right. There is still a lot of, not so much this lack of science when you're doing it quickly, but like there is that moment of a good portion of the work done by your team is in that, let's call it evolutionary like approaches, right? More of that stepwise rather than just get me a number and make it look like a PK profile and get it out the door, right?
Right.
So,
Yeah. So when you're first to deal with essentially that's what the Marines do. They're first to
mhm,
go in, right? So when you're in that position, you are discovering things, just like the
mhm,
name of the team is discovery. They're discovering things, and they're very good at flushing out any you know parameters that or anything that is makes the assay non-robust, and then we will figure out how to deal with it. So that's just one thing that it's not surprising to me that out of all the projects that they've done, they have had some that were unexpectedly finicky, and they were able to get past it and get a good robust assay and pass it on to the regulated team to validate.
awesome. Awesome. We got one more topic here. We got some time for it. Unless we had any last parts about LMPs, otherwise we're gonna ask you guys. Either one of you can answer. It's the future expectations
yeah.
for your
Look into your crystal ball. Where do you think non-GLP LCMS is gonna go?
I think the direction will be doing things that traditionally have been performed by other types of technologies. Not because it's one is better than the other. It's just that everything to me boils down to time and money. How much funds do you want to spend on something, and how much time do you have? And as mass spectrometry shows that we can get to the data at some level fast, and the demands will come. So molecular types is going to essentially extend into the different molecular types, um, and larger and smaller, pushing the envelope on the molecular weight in that direction as well. And I think there will be some level of work that I consider characterization,
Mm,
like a DAR ratio. You measure it
mm,
once, you're done. Unless you change the chemistry, you don't need to do it again. That to me is a characterization. Yes, you're quantifying how many molecules attached. A long time ago, I was working on a conjugation that included a dye agent. And the team thought there was one, and I showed them that no, it's actually average is 2.5. So you have one, two, three, maybe four attached to that big macromolecule. So those are the kind of things that I think will be the future of
Yeah.
non-GLP.
That's great, Habibi, right? Like it is, the sky's the limit for the mass spec. If it's provincial to think, oh, this is a small molecule tool, right?
Right.
That's a provincial thought. And of course, you've been ahead of the
Yeah.
curve for 30 years now with that, right? And then you did touch on dyes, and those are conjugates, right?
Yeah.
And that's really what I don't want to put words in your mouth, but that's what I heard when you said that. And not just the traditional ADCs, it's these arcs, right? And it's even dyes that you talked about, or even like taking a peptide, right? And attaching something to it that's got some business to it, or a payload, right? So those conjugations are really going to be, I think you're spot on with the mass spec side of things there. We're wrapping up. So John, if you want to add anything, or Rochelle, and then we're going to put you two on the hot seat. And I'm telling you, it's on fire today.
Okay.
I think it's like mass LCMS is over the last 30 years or so, particularly since the introduction of electrospray, I think it's still finding what it can do. And I think, as a technology, as it moves into newer areas, then the demand will come to groups like ours. So that's why we've seen that change from discovery being largely small molecule driven to then these mid-size, and then like you say, we have the hybrid team too. It's mass spec is proven to be a good tool that people didn't necessarily know what it could do for them, but it's still continuing to show what it's capable of.
Maybe I have like one follow-up
And just to round that out, and you still need the experts who can apply their knowledge to analyze these more out there molecules and therapeutics and structures, So yeah.
John, I talk about that. The more complex the drug, the harder it is bioanalytically, but more oftentimes it's more effective, right? That's kind of the trade-off. But, I had one little thought before we do put you guys into the fireplace of the seat now. No, what about like the molecular field? So there's like hybridization ELISAs, and I don't know if you know what that is, where you use like some sort of like antisense or something like that to capture, and then we kind of is that being done by mass spec at all? Is there a molecular side here? I guess it's a naive question for me. Is there any like utility in that, or is there not a hybridization type of approach just yet?
Yeah, so I divide the world into essentially what are you measuring and what is the best detector for it, and then the world before that is sample prep. So how do you get your molecule ready for analysis? So it can be almost anything. It could be online, offline. It could be any form of it. And I would say yes. If you think about what is actually being measured, and then say, do we have a mass spectrometer can do that? Basically, the range and the sensitivity and all that. So I think there's room in almost every assay that is not being done by LCMS, that maybe some level of assays or the projects can be done, or it could be a complementary. I think long time ago I think I was talking to you, Dominic, about even the LBA process, ligand binding process, and you get some antibody, a macromolecule purchased from a vendor. How do you know what it is?
Yeah, love it, Habibi. You're right, and we can do on-off rates with things like BIA cores or other instrumentations, but a mass spec would be a powerful tool to characterize it as well. Is that where you were going? I kind of
exactly. So
yeah.
That's the characterization data that feeds right into the rest of the assay. So
Well,
the mass spectrometer doesn't have to do everything. It could have key roles in almost every type of assay.
Yeah, we got this method I was thinking about in another service line. Rochelle, you better take cover because I think I'm going to throw it over to you all because it's very problematic. No, I'm just kidding. Take cover. No. Okay, Rochelle, are you ready to be on the hot seat? We're going to fire away with a couple questions at you. And Habibi, you're welcome to chime in. But of course, it's if you haven't listened to the podcast, I've got to mention pizza. It's mandatory at some point. I got like a big green egg and an uni pizza oven, if you don't know what that is. And I got all sorts of great gear. I love cooking. You don't get this beautiful body of mine without knowing how to cook well. But so what's your favorite dish, Rochelle? What's your go-to favorite dish? Blow us away.
Are we talking about pizza or just like any dish?
It's your favorite. It doesn't have to be pizza. It's your
Oh,
favorite dish to me. Like what makes you the most excited to serve your family or to make? What is that dish that you feel
well,
like I could serve in a restaurant? What's my go-to dish?
I don't know if I would go to, and plus I have kids, so like sometimes like what I desire to eat for dinner isn't always what they desire to eat. So I usually just aim to where everybody likes what we're eating. But I mean, I have a couple of things that I can make really well. Like there's like like a meatball stroganoff thing that I make that's pretty good.
Mm.
I make a good Spanish rice. So
No,
yeah,
nice
things
stuff.
like that. My
So,
kids
yeah,
particularly like
yeah,
both of
it's
those.
better than mac and cheese and chicken strips, right, Rochelle? So your
Yeah,
dishes are you're trying to make something that can satisfy an adult and a kid, and so
yeah,
you make like a beef stroganoff, but you call it a meatball stroganoff, right? So is that or am I putting
yeah.
word? Yeah, wonderful. And then the Spanish rice. Well, just like anything else, tell us a little bit more. You got any secret sauces in there, or secret recipes? Are you using store bought meatballs? You making the meatballs? We want to know.
Yeah, I make myself usually. I try to. I'm probably one of those people who it's like, what do they call it? Like semi homemade, like I use like tricks where I can. Like a lot of times if I have like a chicken dish to like cut on time, I'll buy like a rotisserie chicken and shred that and use that meat instead of waiting for, chicken breast to cook. So,
Amen. I call them street tacos, Rochelle. I recycle all my
yeah,
leftovers. I make kitchen
exactly.
skillets. I make breakfast skillets with anything left over from French fries and things like that. Any potato left over and anything with rice and meat, I make into a street taco. Amen. And I love serving that up.
Good.
Yeah, no, that's great stuff,
So
Rochelle.
do you have to deal with fussy kids in terms of how limited are their diets? Because
Not
I know
really.
we that's you're lucky because we went through years where my kids wouldn't eat much and now they'll eat anything, so it's much easier. And
I
thankfully
mean,
they've
my
moved on.
son My son pretty much eats anything, so I know whatever I cook, he's fine. My daughter's a little more picky, but she's, kind of like expanded her horizons a little bit more as she's gotten older. She's usually the one though that looks at it and she's like, "Ew," and it's like, "But taste it, it's good."
Yeah. And then what about you, Habibi? What's your favorite dish to make?
Well, anticipating the questions you guys asked, I have two. One that I have to go to a restaurant to get, and it's really in this area. It's hard to find one, but it's saffron rice with filet mignon kebab. That's something that I cannot easily make at home, so that's something I would go out for.
Quick
And
question on that one, Habibi. Is it the saffron rice you got to find or is it the saffron? Because I
I
got,
have
you
I
can
have
get
saffron.
saffron.
It's actually the kebab that is.
Oh, the kebab.
Yeah.
Oh, I see. I hadn't thought of that piece.
It
Okay,
has to
great.
be marinated. It has to be right cut of meat, and you can't cut corners. And it's got to, you
I
know,
imagine there's a couple shops down by the 95th and Quivera has a
right.
Mediterranean market there. Yeah, I like that place. And
And
yeah,
growing up on that stuff, it's hard to find a restaurant that actually can duplicate the taste of what you're used to. So you get a little bit of
well, I feel your pain, Habibi. I'm still trying to make Mama's pasta sauce after all 25 years later and thousands of quarts of sauce. I still can't make it like my mom. But
right.
yeah, tell us more.
Yeah. So at home, I can make what my favorite is. It actually is a simple dish: rice and sautéed raisins in it that you mix with it, and you saute it in butter and then mix it with the rice. So I am anything sweet I love, so
Mm,
that it hits it right there. And you can top it with other meats if you
sure.
want, but or you could just have it with the just have rice and raisin.
Yeah, you touched on so I don't use raisins nearly enough. I hadn't even thought of that. Like dates and figs and raisins are great things to throw into. I'm always about me. Of course, you two are talking. I'm going to make it about me. No, the yellow rice. I love making a good yellow rice that I speak with saffron.
Yeah.
Okay, great. Did you have you mentioned both of them? That's awesome stuff. We're going to shift gears into. All right, what's your favorite adult beverage? And by adult beverage, it's got to have some alcohol in it. Okay, who wants to go first? How about you, Rochelle? Tell us what your go-to, I'm going to kick back and enjoy my weekend drink is?
I'm pretty boring. Like I like a good Michelob Ultra, so I'm mostly a
There's
beer
nothing boring
girl.
about that. That's okay. Yeah.
It's ultra, damn it!
That's good. So you like Michelob Ultra? Tell us. Like, is it just? Do you like Miller Lights? Do you like the Bud Light, Coors
Yeah, I
Light?
like I like all of those. I usually stick to the Ultra just because it's a little less carbs, but I mean, I actually like the taste of it. Like it's kind of like got a cool, crisp taste to it that it's good like on a
Yeah,
hot day if
love
you're
it,
kind of
love it. You add
yeah.
any tomato juice at all? I like a good red beer with Michelob Ultra. Are you a red
I
beer?
don't, but I know a lot of people do, but I pretty much just stick to that. I mean, maybe occasionally I might like drink wine or something, but that's pretty rare. Mostly I just drink Michelob Ultra.
Nothing wrong with that.
Uh,
Don't. You should say that proudly. Don't say you're boring at all. That's
Own your tastes,
yeah.
absolutely. Own
Yeah.
your preferences.
And you're not alone. Many people, several of our guests say things like it might be an IPA, but beer is a common answer from people. And
You can't
um,
really go wrong that much with a good beer.
yeah, and you said it's just got to be cold, right? That's the
Yep.
key. And then, especially like you said, as we're winding our way into summer here, right? We get up into that 80, 90 degree, 100% humidity. Man, it doesn't. That's not old fashioned drinking weather, is it? Right? That's a great day to have a good. I like the Miller Lite a little better, but I would never turn down an Ultra. I like that as well. So, thanks, Rochelle. I don't know. Do you chase it with a shot of something or no? You're not a Boilermaker? Or?
I'm not really much for like alcohol, like hard liquor like that. Mostly just stick to, the basics.
No shots of tequila or anything? We're gonna have to get you a Don Field and go
No.
out and get some tequila shots of something. Nothing? No, I'm just kidding. All good. All right, Habibi, tell us about yours. Then we got maybe one more question for you and
I
Rochelle.
will also have a boring answer for you. Actually, my favorite beverage is tomato juice, but the seasoned one that it's, with pepper.
Oh yeah.
It's hot. And it just opens up your taste palette so then the food after that tastes really good.
So is that like a Bloody Mary or a Colmado or in beer or what did you mean by the? I understand what you're saying around the hot, the
Just
spicy.
Bloody Mary mix. No
Yeah,
alcohol in it.
no
Just
alcohol
the
in it.
mix.
Okay. I thought it was a Heineken. Why did I feel like I saw you drink a Heineken one time?
You
Am I
must be
mistaken?
never had never had that.
Okay, it might be somebody else then.
You're thinking about somebody else.
Yeah, that's all right. But no, I do love a good Bloody Mary, but I usually like it, equal parts vodka with it. And it sounds like you're more interested in just the Bloody Mary mix.
So I'm going to chime in here. It's like after doing Venice sort of a month ago, we really acquired a taste for April spritzes, which are perfect going into summer. And with having the front porch built, once that's ready, we'll be christening the front porch with a couple of April spritzes each.
Yeah.
Yeah, it was a drink I had no time for at all, but yeah, totally acquired the taste.
And what about a Negroni? Is that another one you like, John? That's
No, Negroni is still it's that I'm not quite it's still that I'm not quite with the flavor of Negronis yet.
I'm growing into those. I'm starting to
Yeah,
like them. But
yeah.
okay. One last question for you, Rochelle and Habibi. It's just part of our tradition here. We always ask you, what are your weekend plans? And you can talk about the upcoming weekend or whatever might be coming, any exciting summer trips you've got, anything that you want to share with the world, like, hey, I'm going to Europe or going to the Ozarks. But tell us about some upcoming plans you have for the weekend. Rochelle, maybe you can tell us first.
Well, again, I'm going to add to my other boring answer that when it comes to the weekends, I usually look forward to relaxing. And if I have any like chores or anything, it's usually like I'm working in my yard, like, keeping up with my flowers and my landscaping. That's something that I enjoy doing.
Oh,
And that's when I have time because usually my kids have some sport event or something going on. So,
yeah, John's a big gardener. Tell us, I'm going to
Yeah,
ask a follow up to you.
I've got a weekend of thistle digging ahead of me this weekend. I've got a patch that are just gone rampant. So
Yeah, and I did landscaping through high school, and so I do love to garden. I just don't do as much these days. But Rochelle, are you planting some perennials? Tell us what you got going on with your garden.
well, I just got a whole bunch of flowers for my kids for Mother's Day, so I
Ah,
need to put those out in my pots. And then like John, I'm always out there finding some pesky weed. Like I got some choke weed that always comes back and tries to like wrap itself around everything that drives me
Oh,
crazy because I can't ever find the end of it.
yeah. I know what you're
So,
talking about there, Rochelle. They're very deceptive. They look like the plant, don't they? And then they choke
yeah, it
it.
looks like a
Yeah.
vine, and it
They'll-
is, but it's a dangerous one that tries to choke out all your
They
plants.
do.
So
They'll kill your trees almost too. Like I've got some
yeah,
smaller shrubs that if I don't take care of, it'll kill it. And usually you only know it's there when like the fall comes and everything else is brown, and you're like, what's that green thing in there? It's like, oh, well, that's the enemy, right? It's very virus-like, is how I
I
think
don't
of
think
it.
I know its actual name. I just refer to it as the chokeweed.
And it flowers sometimes too if it gets big
Mhm.
enough. It'll actually produce a
It
flower, right? And it just
tries to
kills
trick you.
everything. Yeah, yeah. And so no, that's great stuff. Mother's Day, flowers, all that good stuff. You got a favorite flower, just to follow up? Is there one you really like, is it some
Um,
impatience or nougaty impatience or marigolds,
no,
anything like
really. I do
that?
like marigolds. Those are really easy to grow. And I even had one time where I didn't plant any, so it must have been birds or something like picked maybe like a flower or something and dropped it. I had like random ones grow my front landscaping one year, and that was pretty cool. It ended up being like almost like a big bush of marigolds, but I do like those. Those are pretty user friendly. Like
Yeah.
no one can kill those.
Yeah, and I think they keep the bugs away. That's what I've heard. So I used to put
Yeah, they have
marigolds
a good
around.
smell to
Yeah.
them.
Yeah, yeah, yeah. Okay, great, Rochelle. Thanks. And Habibi, why don't you round us out? And yeah, maybe we'll be wrapping up things here, but tell us a little bit about some weekend plans. I know you're an adventurer. What's going on in your world?
Yeah, so actually this summer, I'm hoping, end of summer, hoping to make it to California to see a close friend of mine. He must be rich. He has some land over there that he's trying to get a pomegranate, orchard going. So I know I've done it before where you're trying to do something by yourself. It goes very slow. So I'm going to go out there and, maybe rest and get a lot of exercise in, trying to help him out with the fencing and adding additional trees, etc.
That sounds awesome, actually. That sounds really cool. Is it Northern California, Southern, Central? Do you know the part?
Yeah, it's in the south part of LA area. It's not in
Oh,
the metro, but
sure.
it's there. Yeah.
Beautiful part of the country.
Yeah.
That's awesome, Habibi. Love that you're gonna get into this. Is that the sunshine? I think Florida's the sunshine state. But
Yeah, exactly.
anyway, they're both great places. But no, that sounds awesome. And you said pomegranate orchard.
Pomegranate.
Yeah. Wow. Fascinating.
Yeah. So
Man.
they're good when they flower. They're nice red bulbs, and they look really nice when they get.
Well, yeah, we're wrapping up here. I got a graduation to go to this weekend. My son's graduating high school. That's got my life pretty consumed. It's been a whirlwind the last few weeks. He's actually wrapping up his lacrosse season too. He's got a playoff game, and earlier in the week we got to walk out in the field with him and stuff like that for his senior night. So that's been exciting. And then I got some family graduating in Boston as well, a high school niece. So I'll be going out there in a couple weeks. That's kind of keeping me busy. But John, you had a lovely trip. I think since the last time we talked, maybe take a few minutes and then we'll wrap it up.
Uh, did I not talk about it before?
I don't know. Maybe you did. I'm losing track.
I think in the interest of time, let's move on
Okay.
and we'll close up because basically it was very quickly. Did Venice, Slovenia would go back to either of them in a heartbeat. They were both fantastic.
And,
In fact, my wife's already plotting how to get back to Venice.
and you've got some grown children. Are you going to Boston or anything this summer? You got some plans to visit them, or what's going on?
Boston in August time, the Chameleons are touring, so I'm going to do Philadelphia on the Thursday night, somewhere in Connecticut the Friday night, and then Cambridge on the Saturday night. So my eldest son and I will be doing that.
Oh, fun.
And then I'm slowly plotting a trip back to the UK in September.
Excellent.
To get together with my sisters and stuff.
Yeah,
But that's it.
you kind of remind me. I will get to see our Lyon site here in June as well. I got that coming up. I'm excited to visit our.
And
teammates in Lyon.
that's Lyon's a great city.
Mm.
It's really nice.
So
Anyway, I think we've run long, so it's probably a good time for us to
yeah,
slowly wrap up. Thanks, Habibi, and thanks a lot, Rochelle, both of you for joining us today. I'm glad we got to explore what your daily routine consists of, and look forward to the next one.
Thank you
Same here.
both.
Thank you for having us.
see you in the funny pages.